Welcome to Dr. B.R. Ambedkar Open University. This is Dhatri and today we are going to discuss about the phylum Platyhelminthes. So, the phylum Platyhelminthes belongs to the kingdom Animaliae and the organisms which belongs to this phylum are called flatworms. They are parasitic as well as free living forms. And more than 13,000 species belongs to this phylum Platyhelminthes. So, now we discuss about the general characteristics of the phylum Platyhelminthes. So, the first one is that they have bilateral symmetry. That means, the organism can be divided into two equal halves. And the second characteristic is they are acoelomates. That means, they lack body cavity or coelom. And the third characteristic is that they have three germ layers: ectoderm, mesoderm and endoderm. So, the outer layers of the organism, they originate from ectoderm, the inner tissues originate from mesoderm and the digestive tract is originated from endoderm. And they have gastrovascular activity, but have only one opening which act as mouth. And cephalization can be seen in these organisms. That means, the nerves and sensory organs are present towards the anterior end of the organism. Now, there are three classes which are present in this phylum Platyhelminthes. So, class 1 is Turbellaria, class 2 is Trematoda and class 3 is Cestoda. So, first we will discuss about the class 1, that is Turbellaria. So, the marine worms and planarians, they belong to this class 1, that is Turbellaria, and they are free living in their nature. So, these planarians, they move using their cilia which is present on the ventral surface or through muscle contractions, and they take up the food, that is the feeding is through sucking action by pharynx, that is the middle portion of the body, and they have gastrovascular activity, but there is no circulatory or respiratory system. So, the exchange of gases is mainly through simple diffusion, and the nervous system of these organisms, they consist of ganglion, that is a simple brain with two nerve cords and an eye spot. This eye spot, it senses the light. And next is the excretory system. So, the unique characteristic of this phylum which have this excretory system is that they have flame cells. So, flame cells, they excrete the fluid waste or liquid waste out of the body. And the next one is reproduction. So, the reproduction of these organisms can be performed by two ways, that is asexual reproduction and sexual reproduction. So, they perform asexual reproduction through regeneration and sexual reproduction is through cross fertilization, because the organisms belonging to this class and this phylum are hermaphrodites. That means, the male reproductive organs and female reproductive organs, they are present in the same organism. And the next class is Trematoda. So, they are called flukes and they are parasitic in nature. So, they are flat, leaf shaped with a conical projection, and they will have oral and ventral suckers and central genital opening, and they have testis as well as uterus in the same organism, and they have the intestines which are bifurcated into two parts. So, here these flukes, they suck blood as well as other cellular fluids. So, they are parasitic in nature. So, the most common disease caused by these flukes is liver fluke disease. So, this involves two hosts, one is sheep and other is snail. So, mainly the primary host is sheep, human beings and cattle, whereas snail is the intermediate host. So, the larvae enters the snail and they starts to grow and develop in the snail, and after they develop in snail they enter the water. So, whenever this sheep, human being or other cattle which take up the leaves from this aquatic lives, that is aquatic plants which are contaminated by this larvae. So, the larvae enters into the host, that is whether it is sheep, human being or other cattle, and they complete their life cycle in these hosts. The other disease which is very common is blood fluke disease. So, this blood fluke is cystosoma. So, here it causes cystosomiosis. So, that is the disease. So, here from an infected human being through feces the eggs enter the water. So, from this egg metaceria larva enters the snail and they escape the snail, and the metaceria larva enters the water, and from water they again enters into the host like sheep or human beings. And the third class is cestoda, and the organisms belongs to this class are called tapeworms, which are again parasitic in nature. So, here these parasites they absorb nutrients, and the cuticle or integument which is present on the surface of these organisms, they make this organism to escape the digestion process in the host, and these organisms consist of scox, that is hooks and suckers, which will help these organisms to bind to the intestinal wall and absorb the nutrients, as these organisms they lack mouth. And proglottids is a segment present in these organisms which consists of the reproductive organs. So, now we discuss about the type study, that is Fasciola hepatica. So, first we will discuss about the history and distribution of the Fasciola hepatica. So, it was the first fluke or trematode that was discovered over 600 years ago, and in 1379 it was discovered by Jehan de Bruyn, and Linnaeus he gave the name Fasciola hepatica in 1758. So, it is large fluke, so which we can see through our naked eye, and it causes liver fluke disease, that is liver rot, which is most common in human beings, but the primary host is sheep, and to lesser extent even other cattle also may get infected by this parasite. So, it is of economic importance because it causes liver rot disease. So, this parasite and the disease which is caused by this parasite is observed in more than 70 countries, including all the continents except Antarctica. So, this is of significance because it is the largest parasite borne disease in human beings, and it is mainly caused in the host, that is human beings, where they live in the areas where the sheep and other cattle are raised, and also in those human beings who eat raw aquatic vegetables. So, it was estimated that over 17 million people are already infected by this parasite and 180 million people are at risk. So, this is the structure and life cycle of the Fasciola hepatica. So, it is flat leaf like structure with a conical projection at the anterior end. So, it has an oral sucker and ventral sucker, and in between these two there is a genital opening, and below this ventral sucker there is uterus, and below that is testis, that is anterior testis, and below that is the intestinal bifurcation, and at the posterior end there is a posterior testis. So, as they are hermaphrodites, that means both male and female reproductive organs are present within the same organism. So, this uterus and testis are present in the same organism. So, these organisms are endoparasites. So, endo means inside, so parasite. So, they live mostly in liver and bile duct of the organism, that is the definite host. And when comes to the morphology, this adult worm of Fasciola hepatica, as I discussed, they are flat leaf like structure, and these oral and ventral suckers. So, the suckers, they usually help this organism to bind to the bile duct wall or liver walls and to absorb the nutrients. So, they have 3 centimeter length and 1.2 centimeter breadth in size. So, it is easily visible with our naked eye, and the intestine is bifurcated. So, it can be divided into 2 halves as they have bilateral symmetry and they are branched. So, once they enter the bile duct, so they live in the bile duct for 5 years in sheep and up to 10 years in human beings. And these are hermaphrodites, that means both male and female reproductive organs are present in the same organism. So, if you see the structure, you can easily identify that uterus and testis are present in the same organism. When comes to eggs, so the eggs are yellow brown in color, whereas the adult worm is in grey or brown color. So, here these eggs are large, ovoid and operculated, bile stained, and they have large in their size, and the egg shells are smooth, which are double layered, and mostly when they are laid they are unembryonated, and the immature forms of these eggs are miracidium, and the eggs from Fasciola hepatica and Fasciolopsis B.Sc. So, we cannot differentiate between these two eggs, they look similar and they appear similar. And the larva of Fasciola hepatica, so it can be in different forms, but the infective form is metaceria larva. So, this actually metaceria larva, which is entering the host, that is sheep or human being, that is actually causing this disease. So, other than this, in between the intermediate host, this larvae can take different forms like sporosis, miracidium, radii, etc. So, now we will discuss about the life cycle of the Fasciola hepatica. So, the life cycle of Fasciola hepatica is complex because it involves 2 hosts. So, the 2 hosts are primarily sheep, human being or other cattle, and the intermediate host is snail, which mainly belongs to the Limnae or Limnidaceae family. And here the life cycle which involves 2 hosts, such kind of life cycle is called as digenetic. and the mode of transmission is that the sheep or the definite host, that means whether it is human being, when they eat the leaves from the aquatic plants which are contaminated by the larvae of this Fasciola hepatica, that is metacercariae larva, then this larvae enters into the host. So, this is the life cycle. You can easily see that through the plants, the larvae, that is metacercariae, it enters into the host. So, once it enters the host, so through the host, through feces, so again the unembryonated eggs are released. This unembryonated eggs, they become embryonated in water after 2 weeks, and then they enter the snail, and in the snail it will undergo different stages, that is sporosis, then 2 cycles of radiae, then cercariae, and finally it escapes the snail and enters the host again. Here this cercariae will encyst itself to form metacercariae. So, this metacercariae form of larva is the actual infective form of the larva. So, how this larva develop in man and sheep? So, first it enters the duodenum, and from the duodenum it encysts itself to form metacercariae, and this metacercariae larva, it enters the lumen of the bowel, and finally it penetrates to the peritoneum, and finally through peritoneum it enters into the liver. So, all these takes place in 1 to 2 weeks after the larvae enters into the host. Then from this liver it enters the bile duct. So, in the bile duct actually it becomes mature adult and it starts producing more eggs, and these eggs now again they are unembryonated because they are freshly laid, and these unembryonated eggs again they enter the intestine and the cycle repeats. And once these eggs are laid, they enter the water. So, where it takes at least 2 weeks at a temperature of 22 to 26 degrees centigrade, where they actually form miracidium, which is the immature form of larva. And this miracidium then enters the intermediate host, that is snail. So, then in the snail this undergoes different forms, that is first they form the sporosis, then they form 2 generations of radiae larvae, and then they finally form cercariae larvae. Then this cercariae larvae, it escapes the snail, enters the water, and it contaminates the aquatic plants. And when they are contaminating these aquatic plants, they encyst themselves to form metacercariae. So, this metacercariae is the actual infective form of the Fasciola hepatica. So, this metacercariae, it enters the host, that is sheep or human being. Again through this sheep or human being, through feces, again this enters the water and this cycle continues. So, the clinical presentation of fascioliasis. So, this disease usually gives some symptoms like fever and hepatomegaly. Hepato means liver. So, the enlargement of liver can happen, and eosinophilia. So, this usually takes some few days to few months of time to show the symptoms. But in some patients, even vomitings, diarrhea, anemia, all these other symptoms also may be observed. And this metacercarial larvae, the migration, it takes 1 to 2 weeks, and usually there are two phases, that is acute phase and chronic phase. So, in human beings mostly we do not find chronic phase, only acute phase of infection is seen, and in the acute phase it takes place like around 6 to 8 weeks, and during this acute phase all these symptoms like hepatomegaly, eosinophilia, then vomitings, anemia, diarrhea, all these kind of symptoms may be observed. And also this worm, it obstructs the bile. So, the bile duct is obstructed by this worm. So, the total duration of this acute infection, it takes for about 6 to 8 weeks. Then it enters the chronic phase where the liver parenchyma is inflated, that means the inflammation can happen. So, the inflammation is nothing but an immune reaction towards any foreign material that is entering into the body. So, this inflammation is characterized by redness, swelling, pain, heat and loss of tissue, that means loss of function of the cells present in the tissue, and it can cause multiple abscesses in the liver. And the obstruction of bile duct, and this causes pipe stem liver. So, this stage is called pipe stem liver, where the bile duct is inflated, the inflammation of bile duct is seen, and this also the final outcome. So, where the liver fibrosis or cirrhosis is observed, and finally it can cause the death of the host. And sometimes the cases of invasion is also seen, that means from this liver or bile duct this worm may travel to the other tissues and other organs, and they infect the other organs. So, here we can see the ectopic presence of this worm in skin, brain and eyes. So, the ectopic means that is other than the original position. So, that means actually the site of infection is liver or bile duct, but here ectopic presence like brain, eyes, skin is also seen. So, how can we diagnose this disease? It is mainly through stool microscopy, that means as this contamination of these worms is mainly through feces, we can microscopically absorb the stool where this operculated eggs can be observed. But after some stage the eggs may not be seen in the feces or stool, and here the concentration techniques like sedimentation is useful, but in the advanced stages this may not be useful. So, we should also look for the other diagnostic techniques. Then the second one is blood picture. So, the another disadvantage of stool microscopy is that the eggs of different species of fasciola cannot be distinguished. So, all the eggs remains similar. So, we cannot correctly diagnose whether it is the disease which is caused by the fasciola hepatica. So, we should go for the blood picture, but in the blood picture we can only detect the whether there is eosinophilia or not. So, what is eosinophilia? It is the increased number of eosinophils. So, eosinophils are the white blood cells which increase in their number only during the period of infection. So, it is like a symptomatic diagnosis. Only if the eosinophilia is present as a symptom, then only we can actually diagnose this infection through this blood picture. And the next one is serological test. So, serological test is nothing but we can detect the antibodies which are raised against this fasciola worm, and we can detect that using the technique like fast ELISA. So, ELISA is nothing but enzyme linked immunosorbent assay, where we usually coat the antigen or antibody. If we know the antigen, we coat the antigen, and or antibody is coated in the well, and we can add the serum sample into that well. So, if the serum sample consists of the antigen, that will go and specifically bind to the antibody which is coated on the well of the plate. So, then it is highly sensitive technique, that is the sensitivity is more than 95% and it is highly specific in nature. So, we can also detect this serological through ELISA, that is serological test, but it can be done only after the 2 weeks of infection. And the other method is molecular method, that is using polymerase chain reaction or PCR. So, in the polymerase chain reaction, we amplify a segment of DNA. So, this polymerase chain reaction, it mimics the DNA replication process which normally takes place in the body. So, here the requirements for polymerase chain reaction are primers which are designed specifically to the segment that need to be amplified, and DNTPs, deoxyribonucleotide triphosphates, then magnesium tag DNA polymerase. These are all required for amplification of DNA, that is PCR. So, here we can amplify the segment of this worm using the probes which are designed specifically for this fasciola worm. And next is imaging, that is using CT scan, MRI or ERCP also we can detect. And we can also use IgE, IgG antibodies to detect this particular worm. And the treatment includes triclabendazole and bithianol and prednisolone. So, these are the 3 tablets which we can use for treating this worm and treating this disease. And finally, how to prevent this disease is by through like awareness of health education, improving sanitation and treatment of infected person and controlling the snails. So, this is all about the Fasciola hepatica, which is a case study which belongs to the phylum Platyhelminthes. So, today we have discussed all about the phylum Platyhelminthes, their classification, and about the Fasciola hepatica and how disease is caused, its habitat, morphology of eggs and larvae and adult form, its life cycle, then treatment and prophylaxis. Thank you.